All right. Can people hear me?
Yes, sir.
All right. Thank you. So we are going deep into the real biochemistry. Um, so this is our third topic, and just to remind you, we have done biophysical chemistry. We have also done lipid chemistry. By now, you are expected to be up to date. That is, you must have answered all both the assignments for the first topic and the second topic. Please, if you haven't done so, do so and submit to my inbox. I hope that is clear. So this is the third topic. So carbohydrate chemistry will be our third topic, and it's something that is very important to know because the carbohydrate metabolism under um, metabolic chemistry is a very, very important component in biochemistry because it carries a huge chunk of the topics in the exam. Okay, right. So we are still doing structural biochemistry. For you to find this class, you have to go into the biochemistry folder, open the structural biochemistry subfolder, then you see carbohydrate chemistry. Okay, so we will now start uploading the notes before the class happens so that people can start going through the notes before they're actually taught. Okay, so just be checking the folders. You see that we'll be sending the notes before the class is happening. Okay, so carbohydrate chemistry will be looking at the structures of various sugars. Okay, the classification of the carbohydrates. We'll be looking at, because this is structural, so we'll be more interested in the structures. Okay, we are not doing any pathways here. We are not doing the glycolytic pathway. We are not doing any other pathways. Okay, we are concentrating on the structures of the carbohydrates. Last week we were concentrating on the lipids. Okay, for today we'll be focusing on the structures of the carbohydrates. Okay, so by the end of this class, would have known what simple monosaccharides are as well as the disaccharides. By the end of this class, would have known what would have known the structures and also the chemical properties of the oligosaccharides as well as the polysaccharides. By the end of this class, you should know the structure and also the function of the sugar derivatives, such as the ones that you saw in PGY 2310 last week. So last week we were looking at the structure of the cell membrane. You saw something that was latent glycoprotein. Okay, so a glycoprotein is actually a sugar derivative. So we'll be very much focused. Our classes are very much focused, straight to the point, and we are focusing on what the MBCHP syllabus prescribes of us to teach. Okay. Last but not the least, we'll be looking at the qualitative and the quantitative analysis of the carbohydrates. Okay. So we start with the importance of the carbohydrates. Why do you think the carbohydrates are important? Anyone? Why do you think the carbohydrates are important? So, what's the importance of the carbohydrates before we even go into their structures and their classifications? Great son.
They are used for energy.
Okay, they are the major source of energy. And we have a very important carbohydrate which is called glucose. It's a Glucose is the major carbohydrate that is required for the generation of energy. in animals, in plants, as well as humans. But as we are doing medicine, so we focus on humans. So glucose is the major carbohydrate required for energy purposes. Okay. What else? That's good. What else? Pauline, what else? Is Wanda in class? Hello, is Wanda in class? Someone else to try out on other laws that the carbohydrate praise.
So for the storing of energy, such as the glycogen is used to store the energy.
Yeah, so glycogen is the major storage form of glucose, the major storage form of energy in mammals, and in this case, humans. Okay, so that's good. What other laws that the carbohydrate praise? Eunice, are you in class? Is Eunice in class? Emmanuel, is Eunice in class?
Yes, she's in class.
Eunice, unmute yourself and tell us other laws of carbohydrates that you think of. Eunice, are you there? Okay, Emmanuel, what else do you think is a role of a carbohydrate? If you are raising up your hand, I can't see you. Today I'm not using my phone. I'm using the PC to teach and also even for the mic. I'm not using my phone, so I can't see anything.
Okay, sir. In Bio 1400, we saw that
Yes.
DNA is made up of glucose, so we can say they are also used to build up other molecules and compounds.
Very good. So that's a very good point that he has brought in. The carbohydrates, they are conjugated with other biomolecules to make um, um, very important molecules like DNA and RNA and so on and so forth. Okay. So the other thing that you can think of the carbohydrates is that they are actually used to make, they are also useful for structural purposes. For example, you saw the cell membrane. The cell membrane is made up of carbohydrates. How much percent of the carbohydrate is in the cell membrane or is in the cell altogether? Let's say how much percent of carbohydrates is in the cell. So proteins are the highest, followed by the lipids. The carbohydrates are at 5%. So carbohydrates, they are also important for structural purposes. You also saw in the cell membrane we had glycoproteins. Those are receptors. So for you to make things like glycoproteins, okay, those things, they require a carbohydrate, okay? They require an oligosaccharide. So an oligosaccharide attached to a protein, okay? So you also have to remember about the structural purposes, okay? So when what comes in your mind when you hear the word carbohydrate? Okay, let's see, Emmanuel, what comes in your mind when you hear the word carbohydrate?
All right. Carbohydrates. Uh, these are components. These are carbo. These are substances. Uh, like that provide energy.
Okay, so when you see the word carbohydrate, the carbo is for carbon, the hydrate is for water, like hydrated. So this is like hydrated carbon. Okay, so carbohydrates, they are comprised of carbon, hydrogen, and oxygen. Okay, so it's a class of biomolecules that is comprised of carbon, hydrogen, and oxygen, and most of them, they have they have different functional groups, like the hydroxyl functional group, the aldehyde functional group, and the ketone functional group. Okay, so where do you think the carbohydrates comes from? These are organic molecules, okay? In organic chemistry, the organic tutor will teach you about the aldehyde functional groups and their reactions. So you should know that carbohydrates are organic molecules which plays an important role in both plants and also animals, okay? And we discussed about the importance of carbohydrates just a few minutes ago. So they are actually structural elements or compounds that are responsible for the functional activities of the cells. And they actually form complexes with other compounds. For example, DNA. Okay, for example, in DNA, you have a ribose sugar which is conjugated with other molecules like the phosphate groups, and also the nitrogenous bases to form either DNA or RNA. So they form complexes with other compounds which are found in the membranes, okay? And I gave you an example of a glycoprotein, okay, or a glycolipid, okay, and also in nucleic acids. Please, this part of the course is meant for the student to understand the structures. You have to be able to draw the structures. Practice makes it perfect. So you have to practice on how to draw the structures. Okay. So there's a simple form of a carbohydrate, and this one utilizes um, it is utilized for the production of energy, and this energy can also be used. Um, it can also be used for the synthesis of other compounds. Okay, so we have simpler forms of carbohydrates that are utilized for the production of energy, for example, glucose, and it can also be used for the synthesis of other compounds in the cell, or it can be used in the case of animals and also in humans for muscular exercise. Okay, so those muscles, they are made up of the muscle cells are made up of structural components that are part of the carbohydrate family. Okay. Okay, so some simple carbohydrate molecules are also components of coenzymes. For example, acetyl coenzyme A is actually made up of a carbohydrate, a simple carbohydrate. Okay, so some simple carbohydrates are also coenzymes. Okay, these are cofactors. They are needed for an enzyme to work well. So we need also understand the chemical characteristics of carbohydrates. we need to know that carbohydrates are made up of carbon, hydrogen, and oxygen, and these are compounds with large molecular weights. We're going to see in a very few minutes. Okay? There are also polyhydroxylated compounds which have at least 3 carbon atoms, okay, and they are actually potentially active. They actually have potentially active carbonyl groups. What we mean by polyhydroxylated, we mean that a compound contains carbon, hydrogen, and oxygen, but then we have multiple hydroxyl functional groups attached to the carbon skeleton, okay, and they have at least 3 carbon atoms. So we begin from the triose sugars going up, triose, erythrose, pentose, and so on and so forth. Okay. So because we can't understand the pathways that are going to come in term 2 without understanding this. Okay. By the way, you're going to have a test just for structural biochemistry. Just for structural biochemistry. So you should start preparing for it
So the
because
test
we
comes
can't
when
proceed
like
to
it comes
metabolic
in
biochemistry.
which which period?
Sorry.
I was saying the test comes when like in which week or which month?
Oh, okay. As soon as we finish this component, you see from the timetable how the topics will be. For example, next week we have protein chemistry. The other week we have nucleic acid chemistry. The other week we have enzymology. So meaning you have, yes, enzymology, then bioenergetics. Okay, that will be the end of structural biochemistry. So we have like 4 weeks to prepare for the test because those 4 weeks we're going to have the 4 topics that I've mentioned. So we need to start preparing for the test, starting from biophysical chemistry, lipids, this topic, proteins, um, nucleic acids, then enzymes and bioenergetics. So it will have like 8 topics or so. Should be 8 topics. Before we go to metabolic chemistry, everyone has to be assessed for structural biochemistry. We see if you are fit to go on to metabolic chemistry, okay? So that we can make recommendations on where you need to touch so well. So we are following the MBCHB curriculum and we are trying as much as possible to maximize the time, okay? So I hope I've answered the question. It will show, so the calendar will show when you are going to have a test. We are going to put the tests on the calendar. So you are going to see
All
them.
right, thank you.
I hope that will be helpful. Okay. So almost all of these carbohydrates they are derived from either they can either be an aldehyde or a ketone. So guys, a carbohydrate can either have an aldehyde or a ketone functional group. Okay, Tutor Ferguson is going to teach you about the reactions of the aldehydes and also the ketones. So you shouldn't worry about that. Okay, so the, we have already mentioned that in their molecules they contain carbon, hydrogen, and oxygen only, unless it is conjugated with other compounds. Okay, so we are still categorizing the carbohydrates. So categorizing just means classification of the carbohydrate. In medicine, what is important is to be able to classify things. If you're not able to classify things, things are bulky and you fail to remember. So for you to remember stuff, you must be able to categorize them, to classify them. Don't just say answers from nowhere. Okay? So the carbohydrates are categorized based on the number of carbon atoms in them. We can classify them based on the number of carbon atoms that they have. Okay? So we can have a simple carbohydrate. Okay? We can have a simple carbohydrate which is made up of 3 to 6 carbon atoms. Okay? And these are called monosaccharides. So simple carbohydrates, they are made up of carbon atoms which ranges from 3 to 6 carbon atoms. Okay. So anything that is in between 3 and 6, it's a monosaccharide. And this is a group which is made up of a molecule very important in humans, okay, and that is glucose. Okay, glucose is very important and it has so many pathways when you start doing the metabolic chemistry. Okay, so two or more monosaccharides can be condensed and they'll give you what is called a disaccharide or a trisaccharide, a tetrasaccharide, or a pentasaccharide. So two monosaccharides joined together, they'll give you a disaccharide. Three monosaccharides joined together, they will give you a trisaccharide. Four monosaccharides joined together, they will give you a tetrasaccharide. Then five monosaccharides joined together, they will give you a pentasaccharide. What is important to remember here is a disaccharide and a trisaccharide. So we have a triose sugar. Okay, we have a triose sugar which is made up of 3 carbon atoms. I'm talking about the simple carbohydrate. We have an erythrose sugar which is made up of 4 carbon atoms. We have a we have a we have a ribose sugar which is made up of 5 carbon atoms. We have glucose or fructose which is made up of 6 carbon atoms. Okay. For the disaccharide, we have we have things like mannose. Okay, mannose is a disaccharide. Okay, we have other disaccharides like um, sucrose. Okay, so sucrose is also another disaccharide. Okay, so the human body does not absorb disaccharides. Remember, you have to break them down to simple sugars, which are monosaccharides. That's where chemistry is based on the monosaccharides and not any other thing. I hope that is clear before we even move on. Okay, so still on the classification. So this table is very important for classification. We have the monosaccharides, we have the oligosaccharides, and then we have the derived carbohydrates. Okay, so one example of a monosaccharide is that you can either classify the monosaccharide as either aldose or ketose. And aldose sugar is a sugar that contains the aldehyde functional group. We have so many mechanisms of aldehydes, okay? So the organic chemistry tutor is going to teach you about those reactions. Okay. Then we have the ketoses. So ketoses are monosaccharides that contains the ketone functional group. We also have the organic chemistry of ketones. So we have mechanisms that will be taught by the organic chemistry tutor. Here we don't teach mechanisms. An example of an oligosaccharide is a disaccharide. Okay. Then we missed one. There's a polysaccharide, which is very important. Polysaccharide is bigger than oligosaccharide. Okay. Polysaccharide and heteropolysaccharide. I want someone to tell us the difference between the homopolysaccharide and the heteropolysaccharide.
All right, for the homo.
Pauline, can you unmute yourself? Okay, can go on. Pauline is going to answer a different question.
All right, for the homopolysaccharide, this means that same kind of molecule, same kind of a polysaccharide, which it's a polysaccharide which has the same kind of molecule attached to it. Then polysaccharide, it's a saccharide which is attached of different molecules.
Okay, to make what Emmanuel is saying clear, what is happening here is that the homopolysaccharide is made up of one type of a monosaccharide. If it is glucose, it will be glucose throughout. Okay. For example, an example of a homopolysaccharide is glycogen. Glycogen is a homopolysaccharide. Okay. Heteropolysaccharide is where you have more than one lactose. Imagine such a polysaccharide. So it will be called the heteropolysaccharide. For the derived carbohydrates, they participate in so many reactions. For example, oxidation can bring about a derived uh, carbo, a derived carbohydrate. Okay, for example, if you do oxidation of a monosaccharide, it will give you something else, which is a derived carbohydrate. Reduction is also another reaction which can give you a derived carbohydrate. Amino acids, when they're being conjugated to a carbohydrate, it will give you something, for example, glycoprotein. Okay. We also have other derived carbohydrates like deoxy sugars. Okay, so those ribose sugar conjugated to nitrogenous bases, those are derived carbohydrates. So classification is very important in medicine, and it will help you have a very good memory, guys. So all simple sugars, just know that they are monosaccharides in nature, and they contain the free sugar group, which can either be an aldehyde or a ketone. Okay, we have already explained what homopolysaccharides are or homoglycans are. These ones, they contain the same type of monosaccharide units. For example, starch, cellulose, glycogen, which is stored in the liver. Okay, we are not talking about animals, we are talking about humans. So glycogen is stored in the liver, and we also the homoglycans, they also contain glucose. Um, so they also contain predominantly glucose. So glucose is the predominant monosaccharides in the homoglycans. And but we have another molecule which is called inulin, which contains fructose. Emmanuel, do you remember inulin from PGY 2310? Anyone who remembers inulin? Gretson, do you remember inulin?
No.
Okay, so inulin is a dye. Remember those dyes we are using the dye method for measuring the body fluid compartments. Remember that? So inulin is used to measure one of the body fluid compartments. Okay. So go back and check inulin. You see that it is, I actually emphasize, I said this is not insulin, it's inulin on the dyes that are used for the body fluid compartments. This one is used to measure should be used to measure the intracellular fluid volume.
So
Yes,
we also need to master it. Inulin, just more like those formalin and others.
yes. Not even the intracellular. It's supposed to be extracellular fluid volume because it does not enter the cells. It's so big. It's a polysaccharide, so it can't enter the cells. I've just remembered. It should be extracellular fluid volume or plasma. So it's either plasma. Inulin it can either be used either in plasma or extracellular fluid, but it must be most likely it's for plasma because it can't even cross those capillary beds. Most likely, inulin is a dye that is used to measure the plasma volume, so the plasma fluid compartment. The heteroglycans, these ones, they contain different types of monosaccharides. Okay, for example, we have hemicellulose, which is a mixture of glucose, galactose, and mannose. Okay, so you have to remember an example of a heteroglycan and say what is in there. Okay, is it glucose, galactose, or mannose? Same thing for homoglycans. Okay, and you have to understand that polysaccharides can contain thousands, hundreds of monosaccharides that are joined by what bonds? what bonds are going to join these monosaccharides together? It's the glycosidic bond or the glycosidic linkage. Okay, so these same polysaccharides can undergo hydrolysis by enzymes or by strong acids. For example, in the gastrointestinal tract, you see how digestion happens, digestion of carbohydrates. So they can undergo hydrolysis. Hydrolysis in biochem means that you are breaking a molecule using water. You are breaking apart a molecule using water. You are breaking that glycosidic bond using water. So the enzymes that can hydrolyze or break down the carbohydrates are called carbohydrates. The ASE is a nomenclature that is used in enzymology to indicate that you are dealing with an enzyme. Okay, for example, you need to remember that cellulose cannot be hydrolyzed by diluted acids, but starch can be hydrolyzed by enzymes. Okay, so starch can be hydrolyzed by enzymes. Okay, so we need to go deep into the monosaccharides because they're very important. Okay, they're the ones that forms the pathways that you'll be learning soon in a few weeks' time. So the monosaccharides can either be classified based on the number of carbon atoms or their activity, their functional group. Okay, so you can either have a triose, a tetrose, a pentose, a hexose, heptoses, and octoses. Okay, so with examples of trioses, we have glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is a very important carbohydrate, it's a very important monosaccharide that is an intermediate in the glycolytic pathway. Okay, it's an intermediate, it's one of the intermediates in the glycolytic pathway, and it is actually produced at step 4 of the glycolytic pathway. You are going to learn all these things. Tetroses, we have a monosaccharide which is called erythrose. Okay, erythrose is one of the intermediates found in the pentose phosphate pathway or the hexose monophosphate shunt. So we're going to learn about these pathways. I'm just telling you so that you know that these sugars we are looking at, they are not just sugars, they are found in pathways. Pentoses, we have a very important pentose which is called ribose, okay, or ribulose. So there's ribulose or ribose. They are two different sugars. Okay, so ribose is a very, very important pentose, and it is actually required in the production of the synthesis of DNA or RNA. Okay, and it is also found in the pentose phosphate pathway or the hexose monophosphate shunt. The other monosaccharides based on the number of carbon atoms are the hexoses. Here, this is where life is. This is where glucose is, guys. So glucose is a major carbohydrate. It's a major monosaccharide which enters different pathways. Glucose can enter the glycolytic pathway and it is going to produce ATP, but the end product of the glycolytic pathway is pyruvate. Okay, so just know that glucose is very important. It can also make the same glucose using a pathway called gluconeogenesis, which is the production of glucose from other non-carbohydrate molecules. So glucose is very important. We can actually make glucose from different compounds, even fatty acids, even proteins, amino acids. Okay, we have different pathways happening in the cells right now, in your cells right now, and they are making glucose or they are utilizing glucose. Okay. We also have um, uh, monosaccharides which are heptoses. Here we have a very common sugar. It's a 7-carbon sugar. It's called pseudoheptulose. So pseudoheptulose is an intermediate again in the pentose phosphate pathway or the hexose monophosphate shunt. Then for the octoses, these are made up of um, 8 carbon atoms, okay, not 9. So you need to understand that all the monosaccharides, whether it's a 4 carbon, 3, 5, whatever it is, it can either be an aldose or a ketose. Okay, right. So you need to remember that the monosaccharides cannot be hydrolyzed into simpler compounds because they are monosaccharides. Okay, so based on the functional group, on the carbonyl group, we can classify them as either aldehyde or ketones, and we call them aldose and ketose. Okay, so we have two molecules in the glycolytic pathway. One is called glycerolaldehyde 3-phosphate, the other one is called dihydroxyacetone phosphate. So one is an aldose, the other one is a ketose. Okay, great son, which one between the two is an aldose? Which one is an aldose between glycerolaldehyde 3-phosphate and dihydroxyacetone phosphate? Which one is an aldose?
No idea.
Sorry, come again.
I don't know.
Alright, the glycerolaldehyde
All right.
is the aldose, then the other one is the ketose.
Yes. The other one is the ketose. Very good. And the one which is an aldose is the one which continues in the glycolytic pathway. The one which is a ketone, it goes into fatty acid metabolism. Okay, it is actually the one used to make fatty acids. Right. That's why they say when you eat too much of carbohydrates, you become fat because of the dihydroxyacetone phosphate, which is diverted from the glycolytic pathway to the fatty acid metabolism. So the aldehyde is CHO. You can see this functional group here. If you see my case right here, okay, right here, this is the aldehyde functional group. So the organic chemistry tutor will teach you the reactions of just this part here. So there are so many reaction mechanisms of this, which you are going to learn. Look at the hydroxyl functional group. There are like 4 of them actually, and this is what we call polyhydroxy. That's why I said carbohydrates are polyhydroxy compounds. This one is a ketose. Look at the ketose. So a ketose, the carbon atom, the carbon oxygen bond is not at the end because this one is at the end. This carbon is just attached to this oxygen like that. Okay. But look at this carbon attached to the oxygen, and there's also another carbon here. If there's hydrogen here, it will turn to become an aldose sugar. It's supposed to be ribose. Okay, it's supposed to be ribose. No, this is deoxyribose, then this one is ribose. Okay. Okay, so you can see my diagram. You can see I've labeled it 1, and you can see that that's the functional group I'm talking about. On the other one, that's the functional group I'm talking about. Okay. So since this is structural biochemistry, they can ask you to draw this sugar. Okay, they can ask you to draw this sugar and label the aldehyde functional group. So you just need to take note. So this molecule here is glycerolaldehyde 3-phosphate. The phosphate group is missing here. It's supposed to be attached. Here. This one is just glycerolaldehyde. Okay, it's a triose sugar. Triose meaning it has 3 carbon atoms. This one is a tetrose and the name is triose. Okay, this one is ribose and it's a pentose. Okay, the one that I actually showed you. This one is glucose, the common glucose. This one is fructose, and sometimes it's called ketohexose because it's a keto. Okay, before we move on, any question? Is Wanda in class? Emmanuel, is Wanda in class?
No.
All right. So, do you have any question?
All right. My question is on the arrangement of the hydroxyl. Now, for instance, you're taught to draw any structure like uh, among those structures.
Yes.
Like, let's say maybe you take maybe one hydroxide to the other side or two of them to the other side, and the other ones like you swap them the way they're looking right now. Then like maybe in terms of drawing you swap them. Is there a problem with that?
There's a very big problem with swapping. A very big problem. It forms a different compound. Okay. There's what we call stereochemistry. So in stereochemistry, the organic tutor will teach you this. You go into the details. In stereochemistry, we do consider the conformation of a molecule in space. So when you
Oh, okay,
swap the
I
hydroxyl
understand.
function group,
The the A1,
it
the
will
D,
produce,
the
yes,
A1 and the D isomers. Okay, all right.
yes. Okay, and to take note is glucose and fructose because they are part of the glycolytic pathway, which is a very important pathway in the human body. Okay, right, and that has been pinpointed for you. So the outdoor sugar and the keto sugar, you can see the functional groups that have been highlighted on the diagram. Okay. For oligosaccharides, we need to remember the disaccharides. These the results from the condensation of 2 to 10 monosaccharide units. Okay. So for a disaccharide, just it contains 2 monosaccharide units, for example, sucrose, lactose. Okay. For lactose, it's very important because there's what is called lactose intolerance. This one is a condition of the gastrointestinal tract where someone who is lacking an enzyme called lactase, deficient in an enzyme called lactase, then they drink a lot of milk. They will present with the uh, osmo, osmotic diarrhea. Okay, they will present with the osmotic diarrhea. Okay, most of the times, if you just drink milk and then you have those bloating and those funny sounds from your stomach, just know that you have lactose intolerance. You are not tolerating lactose, in short. Okay,
But what's the name again? Lactose
because
intolerance or intolerance?
intolerance, lactose intolerance, it's where an individual is deficient in an enzyme that breaks down or hydrolyzes lactose to the two monosaccharides that it is made up of, which is glucose and galactose. It should be glucose and galactose. Yes. So it fails. The person fails. So they have lactose in their gastrointestinal tract, which attracts water and it creates that diarrhea. Okay.
Oh, in short, it's it's that mechanism that we learned in Bao.
Sorry, come again.
In Bao 1400. I'm saying it's the same that it's the same mechanism that we learned in Bao 1400 about the lactose.
Yes, yes, it is.
Oh, all right, okay.
Yes.
Also meaning uh, we're also taught that even Coca-Cola plays an effect. It's more like an inhibitor on that same mechanism.
I'm not sure about that because I haven't come across an article which talks about the relationship between lactose intolerance and
Yeah, Dr.
Coca-Cola.
Shahata once told us to say it's not good to drink Coca-Cola because of the same thing. It acts as an inhibitor on the same mechanism.
Okay, it could be blocking the enzyme and causing lactose intolerance. That would make sense, but I'm going to make a follow up of that. I'll get back to you
Right.
in the group. Yes. Any other questions? Okay, no questions. We move on to the polysaccharides. So these are formed when the monosaccharides are a lot, like more than 10 molecules of the monosaccharide unit. And then they will include things like starch, glycogen, okay, and they have a very important role that they play, which is for structural purposes in cells and also storage of fuel in form of glycogen in mammals like humans. So there's a pathway called glycogenesis, which is the synthesis of glycogen from glucose molecules, and this pathway happens in the liver. Okay, so we're going to learn about that uh, um, in a few weeks from now. Okay, so let's look at the classification of the polysaccharides. We already established that there's homopolysaccharide and there's also heteropolysaccharide. The homopolysaccharides, we have things like starch, glycogen, cellulose, dextrins. Okay, so dextrins, okay, we also have cellular mucoids, which acts as homopolysaccharides. So starch and glycogen, including cellulose, they are very, very important, guys. You have to know the differences and similarities between starch and glycogen, starch and cellulose, cellulose and glycogen. You need to remember the structures because this is structural biochemistry. For the heteropolysaccharides, we have things like glycosaminoglycans, which are made up of amino acids and also the carbohydrate. So things like hyaluronic acid, heparin, which is a drug. Okay, so heparin is a drug, guys. It is used to prevent. It is used to um, prevent blood clotting. Okay, it's a very important drug and it is used to prevent blood clotting. Okay, we have
So I lost the network. You said what? Which prevents blood clotting?
heparin. Heparin is a drug.
Okay.
Yes, and it is used to prevent blood clotting. So we have patients who are um, at um, risk of developing clots in blood, so we can give them heparin. Okay, right. So we have other things like chondroitin sulfate, blood glycoprotein, polysaccharides. Okay, we also have other things like agar vegetable, pectins, hemicellulose. Those are heteropolysaccharides. Okay, for the derived carbohydrates, those that can result from oxidation, we have things like gluconic acid, glucuronic acid, which we are going to see when we do heme metabolism. Okay, which we're going to see when we do heme metabolism. It's a different pathway again in the human body, which has something to do with heme synthesis and breakdown. So we'll see glucuronic acid. Okay, this one is a very, very important group because even in the liver, it has a role which it plays in conjugation of drugs. Okay, glucuronic acid can conjugate drugs and make and render them um, lipid soluble. For example, acetaminophen or Panadol can undergo conjugation in the liver via glucuronide conjugation. Okay? We also have other things like glucalic acid. Okay, it's also results from the oxidation of the normal glucose. For the reduction products, we have things like glycerol or ribitol. So glycerol is a product, it's a derived carbohydrate which results from the reduction of a normal carbohydrate, and it will give you glycerol. So glycerol is also a sugar. Okay, libido is resulting from reduction. We also have amino sugars like glucosamine, galactosamine, mannose-amine. Okay, those are part of the amino sugars. Okay, deoxy sugars, we have things like deoxyribose and ribose. Okay, these are derived from carbohydrates. So all these things we have mentioned here, they are derived from carbohydrates by various chemical reactions such as oxidation, reduction, amination, and also other reactions like condensation reactions, esterification. So right here you can see that the the um, the ribose sugar on the right, on your right, and the deoxyribose sugar on your left. The compound which is on the right is actually
Doc,
used in synthesis of
on the previous slide, are we supposed to gun that table?
let me go back and just see. Okay, this one. Yes, you're supposed to gun. You need to know where each of these sugars is coming from. You need to know, guys. Sugars that comes as a result of oxidation, glucuronic acid. You need to remember glucuronide conjugation in the liver. Okay, so you can easily remember the glucuronic acid. The other one is gluconic acid. The other one is glucalic acid. You need to gun this table. You need to gun it. You need to gun all the other tables. These, you need to gun them. Okay? Structures, you need to remember how to draw structures. This is structural biochemistry.
So in short,
You see,
all the tables that you'll be presenting, we just need to gun them.
you have to gun them.
Short, you need to gun everything.
Yes, but it's easy because you are classifying them. It's not like you're putting things in a disorderly manner. Okay. Any other question? So there will be need for students to know how to draw these structures, okay, exactly the way they are. I'm going to teach you how to do this, okay? Right, now we are done with classification. We move on to something called isomerism, okay? So I have a question for you. What is isomerism? Can you give 5 examples of isomers? Anyone to take up that question?
All right, examples of
Anyone?
isomers.
Yes.
There is optical isomer. There is optical.
No, what is isomerism first?
Or isomerism. I thought maybe you said it's examples.
No, first define isomerism. Anyone? Is Pauline in class? Emmanuel, is Pauline in class?
Yes.
I'm just trying. I'm not sure.
Okay.
Aspiration is when a compound can be drawn up differently, like something like that.
Okay. 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 What is an isomer? What are isomers? Maybe that one would be easy for you. What are isomers?
Uh,
Isomers,
okay, these are substance or molecules of the same structure formula but different in arrangement of molecules. Yeah, of different arrangement in in molecules and all that. Something like that.
Come again. Say it again.
Right. These are substances of the same structure formula but with different in arrangement of its structure alignment and position, something like that.
Okay, so isomers, in a simple way, these are compounds that have the same molecular formula but different structural formulas. Same molecular formula, but different structural formula. We have C6H12O6. Like that's a molecular formula for glucose. It is also a molecular formula for galactose. It is also a molecular formula for fructose. Have you seen that? But they have different structures. That's what isomers are. Isomerism means that it is the existence of two or more molecules that have the same molecular formula but different structural formulas. Okay? Now, on the structural formulas, you can find that you draw them, they kind of look like the same, but when you make the Fisher projection, they'll give you a different conformation. Okay? So, meaning we can further divide the isomers into stereoisomers. Now, we start talking about stereoisomers, meaning we start talking about orientation of the functional groups in space. Okay? Examples of isomers, we have things like glucose and fructose, they are isomers. Okay? Galactose and glucose, they are isomers. Okay? Glycerolaldehyde and dihydroxyacetone, they are isomers. Okay? Right. Is that clear? Okay? So far, I'm sure I can tell me about the classification of the carbohydrates. Someone to just summarize it before we move on to the next stage, where we'll be talking about isomers. Someone to classify the carbohydrates, just in a short summary. A great song. Emmanuel, is Eunice in class? Eunice, can you summarize what we have seen on the classification of the carbohydrates? No one wants to summarize what we have covered. Hello?
Hello, sir, we can get you.
Just do a quick summary. I don't want technical language. Just say whatever you can, whatever you can. We don't need technical language here. Just say what you have learned. So far, me, I've learned this. That's all. Even if you mention one thing, it's okay. We'll move on.
Alright, carbohydrates, these are polyhydroxyl aldehydes or ketones, and then they're
Yes, very
classified
good.
into monosaccharide, disaccharides, uh, trisaccharide,
Oligosaccharides.
oligosaccharide, yeah,
Yes.
and
Tell us about the monosaccharides. Tell
so
us
under
about the monosaccharides.
monosaccharides, uh, we have molecules like glucose, fructose, galactose, maltose. Under disaccharide, we have sucrose, we have lactose, which is very important uh, disaccharides, uh, which is also used in the body whenever there is a shortage of lactose, the enzyme that breaks down the lactose, which is during lactose. I've forgotten the term. Yeah, and then there are also polysaccharides, which are cellulose and starch. We have uh, inulin, we have agar, we have, yeah, that's what I can remember.
Yes, that's very good. You have actually remembered. You have actually remembered most of the stuff, so we can move on. That's very good. Okay, so we have already defined what isomers are. So we need to consider isomers and stereoisomers. Okay, stereoisomers is where you are considering different conformations in space. Okay, that's what we have already defined this, and it will give them different physical properties as well as chemical properties. Okay, so uh, and we need to know what isomers are, what stereoisomers are, and also application of those things here. Now, there's what is called anomeric carbon atom. Obviously, in Bio 1400, you could see questions like alpha carbon, beta carbon. What does that mean? Okay, so um, an aromatic carbon is a carbon which is part of the carbonate group. Okay, in case of an aldehyde, it is at the first. In case of a ketone, it is the second carbon, which is the anomeric carbon. Okay, but why are we learning about anomeric carbons? For what? So we have two conformations because the, the hydroxyl functional group and the hydrogen group attached to the anomeric carbon can easily rotate. And if it can rotate, if the, for example, the aldehyde functional group is facing down, let me go back. If the hydroxyl functional group is facing down, like in this case, this is called an alpha, an alpha hydroxyl group. So this is an alpha carbon. Okay. If it is the other way around, like for example, on this one here, this one, no, not this one. On this one, where the hydroxyl functional group is facing on top, this is called the beta. Okay. So alpha is when the hydroxyl functional group is facing. So an aromatic
Yeah,
carbon is
on
a
those
carbon
two
which
diagrams,
is part of the carbonate functional group.
sorry, so on those two diagrams that
Yes.
you presented, I think there is no alpha there. There is just beta. Are they not betas?
Yes, they're just bitters. I was just giving an example of where you can find the bitter. So the bitter is, you'll see these things on the next few slides. So don't worry about that. Okay. So we have the alpha and the beta anomers, which differs from each other only in respect to the configuration around the anomeric carbon atom. So these ones, it's a type of an isomer. So if I ask you, give an example of an isomer, anomeric carbon isomers. Okay. So we have the alpha and the beta anomers of glucose. Here you can see that this is glucose, right? But then here, this one is the anomeric carbon. On the anomeric carbon here, if you see the hydroxyl functional group is on the on the on the left, like in this case, on the left means it's down, okay? On the left means it's down. Whatever is on the left, when you go in the chair, the Fisher projection, sorry, this is a Fisher projection. When you go in the chair conformation, you see that the hydroxyl functional group will be down, okay, will be facing down, which means that particular hydroxyl group is alpha. So we say alpha D-glucopyranose. Okay, I'm going to teach you what these things are. So the D is the mirror image. If you put a mirror here, you're going to see this compound on the other one. Okay? No, not these ones, not these two. These ones are both D's. We have D and L conformation. So when you put a mirror, you see one image on the other one. That is D and L isomers, okay, or optical isomers, okay, because when you pass light through them, they're going to divert light either on the left or on the right. That's why I call D and L conformations. So D and L, we also have alpha and beta. When you see the hydroxyl functional group, so this one is supposed to be on the left like that, on the right, sorry. So this is on the right, on your right. So on your right, it means alpha carbon. It means it's down like that. On the left, it means it's on top, and that is going to be beta D glucopyranose. Forget about the pyranose thing. I'm going to teach you in a few minutes. Now, there's what we call cyclization. Okay, you have to show this in the exam. So there's where you need to show the cyclization, because these carbon, these sugars, they don't exist like this in real life. You can't find glucose right so like this, or glucose in this form. No, you can't find it. So there's what we call cyclization. Cyclization requires an, um, an a neutrophic attack of this, the hydroxyl functional groups attacks the carbonyl group here. So when that attack happens, this molecule opens up, and you see what happens. The next thing that happens, the oxygen is attached like that, like in this case. Okay, so this oxygen attacks this. Then once it attacks this molecule, what will happen next is that this hydrogen has to leave. Okay, this hydrogen has to leave. When it leaves, that's when this oxygen will be free. Okay, so there's this reaction. It's actually an organic reaction. Okay, so the organic chemistry I don't teach you very well. I don't teach organic chemistry. So this oxygen attacks the carbon which is here, and this oxygen opens up, the electrons opens up like that, and it's the one which becomes hydroxyl. So this hydrogen which was here initially, it will be grabbed and then it will be taken to this oxygen, so that you have the hydroxyl functional group there. So you can either have this conformation where the hydroxyl is facing up, or another conformation where the hydroxyl is facing down. Okay, so this is also called a chirocarbon. Okay, chirocenter. Chirocarbon is the same thing as anomeric carbon, guys. So this is an anomeric center. If they ask you how many chirocarbons we have, or how many anomeric carbons we have, so the anomeric carbon is this one. Okay, so chirocenters. So this one is chirocenter, which has been formed. Okay, so chirocarbons are those carbons which are attached to four different groups. For example, this is a chiro This is a chiral carbon. This one is a chiral carbon. This one is another chiral carbon. This one is another chiral carbon. And this one is another chiral carbon. No, this is not a chiral carbon. It is attached to similar atoms. So this one, no. So in this molecule, we have 4 chiral carbons. 1, 2, 3, 4. This one is not a chiral carbon. Okay. But we have only one anomeric carbon. Please, these things, they come in the MCQs. So you have to pay attention. Which one is the chiral carbon? How many chiral centers do we have in ribopyranose? It's just 4. Okay. Right. So the other conformation will give you an alpha D glucose. Okay. 1, 2, 3, 4, 5. No, it's not supposed to read glucose. It's supposed to read alpha D ribose. Okay. This is not glucose. Is this glucose? No, this is not glucose. It's ribose. So these ones are supposed to read as beta D ribose. Okay. The ones which are down are the ones which are supposed to read glucopyranose. These ones, yes. So something happens with the carbonate group here. Okay, this is a reaction. You have to show this reaction. Okay, they will ask you, show this reaction. So this oxygen attacks here, and this oxygen opens up like that. And then the next thing you're going to show is here. Okay, the next thing you're showing is here. So there's alpha, both alpha and beta, and you have to give both ways for alpha and also for beta conformation. Please remember the thing about the chiral centers. Glucopyranose has got 6 chiral centers. Okay, 1, 2, 3, 4, 5, 6. Okay, that is cyclization. Any question before we move on?
Dog, can you repeat on the difference between the chiral and the numeric centers? I don't know if it's a numeric.
So an anomeric carbon is a carbon which is attached to either an aldehyde functional group or a ketone functional group. That's an anomeric carbon. A chiral carbon is a carbon which is attached, which has 4 different atoms attached to it. It can also be an anomeric carbon anyway, but an anomeric carbon is just one attached to either an aldehyde or a ketone functional group. Okay, so I need to edit the notes. This thing has to go down here. Let me do it now. I need to edit the notes because they will be confusing to people. So this one is beta and this one is alpha. Okay, I think now we are safe. Let me put it here. Okay. Any questions? You have to learn how to do this on your own. Okay. In first year in Bio 1400, we taught you about how to draw these. So you are expected to know how to draw them. You need to know that here the hydroxyl group is on the right, here it's on the left, here it's on the right. They're alternating like that. You also have to learn how to do the cyclization. They do ask, it comes in the test. Okay, you have to learn how to do this. Okay, go into the epimers. Anyone who can tell us what epimers are? Anyone to tell us what epimers are? Emmanuel, what are epimers?
Uh, like if I could remember very well, these are molecules of the same structure formula, same molecular formula, but only different in uh one particular position. Yeah, in one like
Yeah, can you give?
e.g. glucose and galactose. Oh, yeah, glucose and galactose because they just differ in carbon 4. The other one is on the right, the other one is on the left.
Okay, so in short, you are saying galactose is a 4 epimer of glucose, meaning if you know how to draw the structure of glucose, definitely you know how to draw the structure of galactose. What Emmanuel is saying is that here at carbon number 4, just swap this hydroxy, take it on the other side, the hydrogen on the other side, everything else should remain intact. Okay, do we have anyone who has a problem? So epimers, these are isomers where they only differ at one position. Okay, everything else is the same, but they only differ at one position. That is the conformation on one position. So the monosaccharides which differ in the configuration around one specific carbon atom are called epimers of one another. Okay, we have C2 epimers of um, glucose and mannose, which means mannose is a 2 epimer of glucose. If you know how to draw the structure of glucose, definitely you know how to draw the structure of mannose. If you know how to draw the structure of glucose, definitely you know how to draw the structure of galactose. You don't have to memorize each and every structure in biochemistry. You have to be very clever and you have to be on point because you are reserving your memory for very important pathways which are coming. Okay, so we are here, beautiful diagrams. So this is D glucose and this is D mannose. Okay, right here you can see the hydroxyl functional group is facing on the left, on your right, sorry. Here on the other hand on D mannose, it's on the left. So meaning everything else is the same except this. And you see that this has got consequences in the cell. Why is it that the human cells they can't take up mannose into the glycolytic pathway? Anyone to answer that question? Why is it that the human cells can only accept this molecule into the glycolytic pathway and not this one? Pauline, can you answer that?
Sorry, I didn't get your question.
Why is it that the human cells they are unable to take up D-mannose in the glycolytic pathway? Why is it that they can only take up glucose, even if they only differ here? Why is it that the the pathway can only take up this molecule and not the other one? Why?
I'm not sure.
Yes, Pauline. Okay. Great son.
Into my mind is the alpha linkages. Maybe with the dimanose, we have the beta linkages, which maybe won't be accepted.
Okay. Okay. When we draw this molecule, where is this carbon going to be? What is going to happen to what is going to happen to this hydroxyl group? Is it going to be, because in a normal case, the second carbon, the hydroxyl group is facing down. In the other form, this one will be facing up. Okay, this one will be facing up in that chair conformation. Now, it is a very simple question. There's no enzyme which can add a function, which can add a phosphate group on the 6 carbon here when when the structure is in this form. There's no enzyme. In short, there's no enzyme for this substrate to add the phosphate group here. But there's an enzyme which is required, which adds a phosphate group right here at carbon number 6. That's the reason why this molecule cannot enter in right. There you can see the difference. Okay, we have another one which is galactose. It's a 4-epimer of glucose, and you can see what's happening here. Okay, the D-glucose, the hydroxyl functional group is on the right, whereas on the other molecule, everything else is the same. Take note, guys, this is the Fisher projection. Take note that everything else is on the right. Everything else is normal, but right here on carbon number 4, the hydroxyl functional group is on the left. Okay? So, again, this molecule cannot enter the glycolytic pathway. Only this molecule can enter the glycolytic pathway, nothing else. Okay? Okay, now we are going into what is called, so here you should
Yeah.
get knowledge of
Me,
D and L.
my network was bumping. I just want to get a clarity on why is it that manners it can't enter into the glycolytic pathway.
It's because there's no enzyme for this substrate. There's no enzyme for D mannose.
So
Okay,
in short, even
that's
for
why we
galactose,
don't
there's
have
no enzyme for it.
enzymes. Even for galactose, there is no. Yeah, There's no such a thing. We are talking about humans. Remember, we are in medicine. So we are restricting ourselves, humans. When I say we have no enzymes, I mean we have no enzymes in the human cells. Okay? Okay, we can move on. The next thing. So now we are talking about the reactions. Remember, we we need to understand the reactions of these carbohydrates. Okay, but Tutor Ferguson is going to go deep into the reaction mechanisms. Here, no, we're not going to do that. Okay, so we have what we call the reducing and the non-reducing sugars. Okay, so what are reducing sugars first? Maybe someone can help us. Is is um, is Eunice still in class?
Yes.
Yes, sir.
Eunice, can you tell us what reducing sugars are? Hello, Eunice. Okay, she doesn't want to speak. Pauline, can you help us with what reducing sugars are and give us a difference between a reducing sugar and a non-reducing sugar? Pauline, can you unmute yourself?
A reducing sugar is a sugar that it donates electrons to different compounds.
Okay, okay, that's a good try. Great son, can you polish up what Paulina said about the reducing sugars?
Not very sure, but a reducing sugar undergoes oxidation, meaning it's able to act as a reducing agent.
Okay, okay. Give an example of a reducing sugar.
Sucrose.
Okay, so this is chemistry, guys. This is
Okay, out
chemistry.
of reducing sugar, they easily dissolve, and for
No, no,
non-reducing,
no, no. No, 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 no. We're talking about reactions. So we need to talk about reactions. This is chemistry. So which part of the reducing sugar molecule is responsible for reduction? Which part of the reducing sugar molecule is responsible for the reduction that you're talking about? Reduction is where electrons move from one molecule. They are lost. In short, the transfer of electrons from one molecule to the other. Once you just lose the electrons, that is reduction. So once you gain electrons, that is reduction. Okay. So electrons are being transferred from a reducing sugar to any other compound. It could be a metal. Okay. So we need to talk about the chemistry. Which part of the reducing sugar molecule is responsible for the reduction? So it causes reduction of another molecule. That's why it's a reducing sugar. So which part of the molecule is responsible for the reduction of other compounds?
at the carbonyl group.
The carbonyl group. Exactly. So without the carbonyl group, no reduction will be taking place. Okay, because that oxygen which is attached there, it has got lone pairs of electrons, and those lone pairs of electrons, they are able to undergo transfer. Okay, they can be put off if you have a powerful oxidizing agent. For example, if you have a metal copper, so copper 2 sulfate found in the Benedict solution is able to pull the electrons all the way from the reducing sugar molecule to itself. Okay, and then it will undergo reduction, change the colors. That's the chemistry that we wanted. Okay. Okay, so when you gain the electrons, that is reduction. So a molecule that gains an electron is therefore called what? An oxidizing agent. Okay, or it is reduced. So that molecule is reduced. A molecule that donates electrons is called a what? A reducing agent. Okay. And a sugar that donates electrons is called a reducing sugar. Okay. And you need to understand that the electrons are donated by the carbonyl functional group, which Emmanuel just mentioned. In the Benedict reagent, the changes that you see when you expose that molecule when you expose the copper 2 sulfate, the reagent to a reducing sugar, is actually it's actually the reduction, the redox reaction. Okay, I hope everyone still remembers what they did in the Bio 1400 lab about the reducing sugars. So that experiment we gave you guys, it had four things in it. There was test for reducing sugars, test for proteins, lipids. We gave you that thing. and you have to know that those color changes, they happen because of the electron transfer. Can someone just tell us the color changes all the way from green? Is it from blue? Yeah, from blue. Someone to tell us the color changes. Hello? Great son. Tell us the color changes. You missed that lab.
We use the clock.
Or use crooks. Even you, Pauline, you use crooks.
From green, orange to purple. Oh,
Hey, which one is that?
to
Can I
yellow
try?
or to orange to red.
Pauline, yes, you can try. There's even red. Pauline, you can go on.
So it's blue, green, yellow, orange, and brick red.
Yellow, orange, and brick red. Yes. So that's what is happening. The color changes, and they happen because the higher the amount of the reducing sugar, the higher the deeper the color. Okay. So this molecule here, one, two, this is glucose. So this is glucose, guys. Okay, this is glucose. So you can see here on the carbonate functional group, this oxygen here is the one that donates the electrons. Okay, donates the electrons and then it becomes oxidized. Oxidation happens to this molecule. Okay, what changes the color is the oxidizing agent, the copper 2 sulfate. Okay, here you are, we're in the lab. So with water, you see this blue, nothing is happening. With glucose, you can see that dark blue, the brick red color. You can see with onion has got a reducing sugar. You can see, but not a lot. Okay, there's this dash which is broken down there. So you can see the orange in potato. You can see at least the yellow, green. Okay, starch. There's nothing. Okay, nothing. There's no reducing sugar there. In onion, could be that there's fructose, which is the reducing sugar for in the fruits. Starch, nothing. Okay. Right. So you need to remember these are qualitative tests. In fact, they can also be quantitative because the deeper the color like this, you know it's a lot. There's a lot of reducing sugars. Okay, so the Benedict reagent, it undergoes a complex color change when it is reduced. Okay, and you saw this in the lab. Okay, so the intensity of the color is actually proportional to the what? To the concentration of the reducing sugar. Okay, someone has mentioned this. Pauline mentioned the sequencing: the blue, green, yellow, orange, brick red. Did I just leave the class, or there's someone who just entered? Did I ever leave the class at any point? Hello?
No, there's someone who just left.
Or someone who just left? Okay.
Actually, two of them now. We're now remaining two of us. Three.
Okay.
Pauline, me, and you.
Oh, okay. We are remaining the two of you in short. Okay. So um, we have already learned that the aldehyde functional group is the one that is responsible for the uh, for the reduction that we see. Okay. And now the conformations now. This one is alpha glucose and the other one is beta glucose. When you see the hydroxyl group on top, then you know it is beta glucose. You see it down, then you know it's alpha. You need to know how to draw the chairs, okay? Very important. Okay, so they alternate the hydroxyl down, the hydroxyl up. So whatever you see on the left. Okay, so you have to take note that the carbonate group is very free in the stretch chain form, but it is not free in the ring form. So it is not free when you have a ring like that. But you have to remember that this ring can form and also the stretch chain form can be interchangeable, meaning it can rearrange, go back to the stretch chain form, which is the Fischer projection. In short, all monosaccharides are reducing sugars. Okay. Okay. So sucrose, is sucrose a reducing sugar?
No,
No. Why?
because it doesn't have a free carbonyl group in the ring chain. It doesn't have an open, yeah, something like that. It can easily react.
Okay, so it doesn't have the free carbonate group which can react. So that's the reason why it is not a what? A reducing sugar. Please know how to draw the structures of the disaccharides. Please, please, please know how to do that. Practice will make you perfect. Okay. So the answer is that they have no free carbonate groups, and these are called non-reducing sugars. They do not reduce the Benedict reagent. Okay. Okay, so we are talking about the same thing. So for you to know, you can actually test for the non-reducing sugars. You can hydrolyze the sucrose. It breaks down into the normal sugars, the fructose and the glucose, and then what do? You test if it's going to turn brick red. Then you know in the first place it wasn't a reducing sugar, but after hydrolysis it has become a reducing sugar. So you do hydrolysis with an acid. Okay, so now we move on to the very important carbohydrates that we need to remember. Okay, so these are monosaccharides, and we have pentoses like ribose, a very important carbohydrate. This one is found in the nucleic acid. You know it's found in RNA, and we have forms of structural elements of nucleic acid and also coenzymes. So this same ribose forms a coenzymes. It is also an intermediate of the pentose phosphate pathway. I already mentioned this. Used to make ATP, NAD, NADP, and flavoproteins, which are actually coenzymes. If you have your mic unmuted, please mute it because we are recording the class. The person who is having their mic unmuted, please mute yourself. Other sugars that are very important, we have glucose, which is found in fruits and juices, and upon hydrolysis of starch, you can also have glucose, maltose, and also lactose. Okay, so the body sugar and also the principal one, which is actually used by all the tissues in the body, for example, the brain, the kidneys, and all other cells. Okay, and an excess of glucose will be give you a condition called hyperglycemia. It's a very bad thing. Okay, and also the presence of glucose in urine is called glucosuria. So when we check your urine and then we find there's glucose in your urine, we call that glucosuria, and it is a problem. It indicates that the person might have diabetes mellitus. Okay. It is also due. It also causes cataract formation, which is due to sorbitol, which contains glucose. Okay, cataracts. It's a condition in the eyes. Okay, it affects the eyes and it causes clouding of the lens, and it leads to damage to the what? Damage to the eyes, and which leads to blindness. Okay.
So
Another
a question.
yes.
So for those that have glucosuria, like they have dark, their urine it's too dark or what?
No, it's not even dark. It's just normal urine. But when you test, you find that there is glucose in it. In short, when you do that reduction test of the Benedict test, you see that the urine is giving you an orange color. Then you know that there's glucose in here, or there's a reducing sugar in here. Most likely glucose.
Also, meaning even in labs in hospitals, like they use Benedict as well, just like we did in labs,
Yes, they use Benedict.
right?
Yes. Okay, so the fructose is another important sugar and is found in fruit juices, in honey, and it is actually released by hydrolysis of inulin. Okay, remember inulin is a polysaccharide. It's actually the main nutritional source of energy for the spermatozoa. So the sperms, they use fructose, not glucose, guys. So the major nutritional source of energy for the sperm cells, is fructose and not glucose. And it is actually found in the seminal fluid in the prostate gland. Fructose can Take note that fructose can be converted to glucose in the liver, and it is actually the sweetest sugar that we have. Okay, and if you don't have an enzyme which can break down fructose, it will lead to a condition called essential fructosuria. Essential fructosuria is a condition where someone is urinating fructose. Okay, their urine is composed of fructose because it's a lot. Another important carbohydrate is galactose, sorry, and it is actually found as a component of lactose in milk. Okay, it is also formed by hydrolysis of lactose, and it is actually synthesized by the lactating mammary glands. So someone who is breastfeeding, they are producing lactose. Okay, it is a constituent of the glycolipids and also the glycoproteins that we learned in PGY 2310. It can actually be converted to glucose in the liver. So the hepatocytes can convert galactose to glucose. It can actually accumulate, and its accumulation in the body, it can lead to a condition called galactosemia. Galactosemia is a condition where you have excess galactose in blood. Okay, and it is actually implicated in cataract formation because of the galactitol, which is um, an alcohol form of the galactose. So it can
So,
lead to
sir,
cataract formation.
a question:
Yes, please.
whenever a condition it has same meaning or or
Emia
maybe at
means
the
in blood.
in blood,
Emia
right?
means in blood. Urea, like urea means in urine. Okay, so if I say galactosemia, I mean increased galactose levels in blood. If I say galactosuria, it means increased concentration of galactose in urine.
All right, thank you.
You're welcome. So there's another thing which is called the glycosidic bonds. So here you have to remember the different types of glycosidic bonds. You can either have the alpha glycosidic bond or the beta glycosidic bond. Okay, based on the position of the carbon atom hydroxyl functional group, which we already know. You already know about this. Okay, so alpha bonds, they are like this. You see, they are like this. For the beta ones, for the beta ones, they are like this. Okay. Okay, so these are beta bonds. Okay, based on the position of the hydroxyl functional groups. Okay, take note here. So the linkage between the C1 and alpha hydroxyl group and the difference here. The linkage between the alpha, the carbon 1 beta hydroxyl and a C4 hydroxyl. So again, C1 and C4, you can see the positioning of the hydroxyl functional group, okay, as either alpha or beta. Okay, so you are experts in that. So the C4 end can either be up or down depending on the orientation of the monosaccharides. Okay. So take note that we are talking about the C4, C1 and C4 in relation to the alpha glycosidic bond. Here we have carbon number 1 going to carbon number 4. Or carbon number, again, carbon number 1 beta, carbon number 1 going to carbon number 4 the hydroxyl. So this one, the other one would be called the beta, the other one would be called alpha. Okay. Right. We also talk about oligosaccharides. Okay. So we have sucrose, which is a disaccharide, and it is actually table sugar. Okay, from the caffeine sugar. So the alpha D glucose and the beta D fructose will give us sucrose. So remember the sugars that gives you sucrose, guys, and be able to draw. Okay, so remember there's alpha D glucose which starts first. When you swap them, it will be incorrect. Putting fructose in front is incorrect. You can only press them like this. Okay. So my question is this a beta, is it an alpha glycosidic bond or a beta glycosidic bond?
Edward,
Emmanuel, is this an alpha glycosidic bond or a beta
it's
glycosidic
an
bond?
alpha.
Okay, yeah, so this one is an alpha. Okay, so this one molecule is called alpha D glucopyranose, glucopyranosal. Then we indicate the 1,2, okay, beta D fructofuranoside. So the pyranosal indicates that it's a pyranose ring six-sided molecule. The furanose ring is five-sided molecule. Just like you can see here. You can see this is six-sided, this is five-sided. Okay? You have to remember this name. Alpha D glucopyranosal 1,2 beta D fructofuranoside. Okay. Draw the molecule and make sure that you know the glycosidic linkages. The size by the lactating mammary glands, meaning someone who is breastfeeding, they are producing lactose. Okay, and you remember the condition galactosemia. Okay, sometimes someone can abort a baby, but the breasts are still producing milk. Okay, which leads to discomfort. Someone is producing milk, but they don't have a baby. The baby died. Just imagine. Okay, so some condition may give you the galactosemia, where you have the excess galactose in the blood. So this one is milk sugar, okay, and it is made up of beta D galactose combined with beta D glucose in that order. So there's B1 to there's B1 to 4 linkage. So this one is a B1 to 4 linkage. The first one was the alpha 1 to 2. So one alpha, 2 beta. So the enzyme lactase and also lactose intolerance is very important, as you can remember just from the start of the class. Okay, so this one is the least sweet sugar, about 16% of sucrose. And it actually possesses one potential aldehyde functional group. Okay, so it when it breaks down, It's actually reducing sugar, and it forms the ozazone crystals when because of the aldehyde functional group. For the homopolysaccharides, we need to remember starch, glycogen, and cellulose. So starch is the main storage form of glucose in plants, and it is made up of the glucose um, alpha glucose, D alpha glucose sugars. So remember that it has got two forms. You can either have amylose or amylopectin. You need to understand the difference between amylose and amylopectin. Okay, so 20 to 28% in the plants is amylose. Amylopectin is 72 to 80%. It's more than the amylose levels. So now you need to know the difference between the two. There's one which is branched, the other one is not branched. So this one is a polymer. These two are a polymer of alpha D glucose. You have to remember that point, guys. Very important. Okay. So we have the alpha 1 to 4 glycosidic linkage, and we also have the alpha 1,6 linkage in the same molecule. Okay. We don't have any free aldehyde group, and that's why it is not a reducing sugar. Okay. It is found in wheat, rice, corn, potatoes. Okay. So you need to know the differences between amylose and amylopectin. Okay. Which one is water soluble? Which one is more relatively water soluble between amylose and amylopectin?
Amylose.
Hello? Yes. So here we have this diagram here. Before it even come there. So there's 1,4 glycosidic linkage here, right here, and there's also 1,6 glycosidic linkage there. So if you're asked to point at those functional groups, you need to point at them and remember they are very important there. You're going to use them to differentiate starch from any other molecule. So amylose, now the characteristics now. So the amylose exists as a in linear form, whereas the other one, oh, in linear form, whereas the other one is branched. So you need to remember the differences between amylose and amylopectin. Amylose linear, amylopectin branched. It has got branches. It's the one which has got the 1,6. Amylose just have 1,4. Sorry, some 1,6, some not everywhere. But for this one, it is alpha 1,14 or 1 to 6. Okay. So remember, this one has mostly just one alpha 1,4. Okay. The polymer units, this one is smaller. The amylose is smaller. It's like 200 to 2,000 glucose. This one is above 2,000. Okay. Above, in fact, it's above 2,000 up to 2 million. Okay. As usual, you can see that the molecular weight, this one has to be less than 0.5 million. This one has to be between 50 to 500 million. Okay, this one does not, this one, the first one forms a firm gel. Okay, the other one does not form a gel. Okay, you can see these are amylose. The other one with the 1,6, the branches, it's amylopectin. Please, when we start metabolic chemistry, we are not going to start talking about structures. This is the only time we are going to start memorizing the structures. Then we'll start talking about structures. Okay. Then we also have another sugar which is called glycogen. This one is very important. Okay, very, very important. Don't forget cellulose. So this one is very important. It's the major storage form of glucose in animals, in humans. Okay. So it is actually known as animal starch, and it is present in the liver and also in the muscles. Okay, because there's glycogenesis which happens in the liver and also glycogenesis happening in the muscles. So the glycogen, it has both alpha 1, 4 and alpha 1, 6 linkages that are found in its structure. Guys, this is very important because we have different enzymes that can act on the alpha 1, 4, and also we have another enzyme which can act on alpha 1, 6 when you are breaking down the glycogen in glycogenolysis. So there's a pathway called glycogenolysis, which is the breakdown of glycogen to the glucose molecules. It's actually much more branched than the starch. So already you can see the differences between glycogen and starch. It is much more comparable to amylopectin than amylose. Okay, and it actually gives a red color with iodine. Okay, so this is a structure of glycogen. It looks more like it's amylopectin. Okay. So someone to tell us the difference between glycogen and amylopectin anyway. What's the difference? Pauline, what do you think is the difference between glycogen and amylopectin? Pauline, are you in class?
Yes, sorry, please repeat that.
What do you think is the difference between glycogen and amylopectin?
I'm not sure.
Emmanuel?
Alright, for the amylopectin, the difference is the branching. The glycogen it is more branched than the amylopectin.
Yeah, yes, that's very true. Okay. So they both have 1, alpha 1, 4 and alpha 1, 6. Okay. Do you have any questions? This one was a nice class. We need to upload it on YouTube. Do you have any questions? Any question? Pauline, do you have any question?
When it comes to writing notes about carbohydrates, do we also need to draw the structures?
Yes, you need to draw the structures. This is structural biochemistry. Let me go back to the first slide. This is structural biochemistry. So the first test that you are going to have. So here it will be like this. Before you have any test at school, we are going to be having but we are going to have 3 tests for this course. 3 tests for PGY 2310 and 2 tests for PTM. We have 2 tests for anatomy. We have 2 tests. What's the other course? 1 test for psychology, behavioral sciences, and we have 2 tests in organic chemistry. Okay, so you are going to see the format. You see from the test that you are going to write, you see the format for this for test 1 is supposed to be MCQs for this course, MCQs and short notes. So you see, Pauline, this is structural biochemistry. So the structure is not for fun. It's actually, it means structure, meaning you are supposed to draw. You need to start practicing drawing. Are we together?
Yes.
Yes. Um, Emmanuel, Emmanuel, are you in class? I'm giving you a chance to ask me any question that you want. People have no questions? So we are remaining with at least 4 topics. Let's just count them together before the test. So there is protein chemistry, there is nucleic acid chemistry, there is bioenergetics, and there is enzymology. 4 of them. So we have 4 weeks from now to start preparing for the test. The format will be MCQs and um, short answer questions. Okay, if people don't have any questions, we have an assignment which will be uploaded in due course for this topic. Every topic has an assignment. There is no chance to rest. Kindly submit the assignments if you haven't done so.
All right, sir.
Maureen, are you able to see the assignments?
Yes.
Okay. So let's just stop recording and we stop the transcription.